In this video, we're going to be talking briefly about all the different kinds of transport that occurs across a typical cell membrane and the basic mechanisms thereof. What the others have to do with it is that they're there to lull the viewer into a false sense of cuteness. So, without further ado, Irina B. and Linda Phillips would like to present cell membrane transport. If plasma membranes had been composed purely of lipids, only select molecules would have been able to successfully diffuse across them at the rate necessary for the cell's normal function. Examples include small hydrophobic molecules such as O2, CO2, N2, etc. On the other hand, small uncharged polar molecules like water and glycerol would only be able to diffuse across the plasma membrane much more slowly, with large uncharged polar molecules being able to diffuse only at a virtual crawl. As for ions, a purely lipid plasma membrane would be virtually impermeable to them. Fortunately for living things, however, 50% by mass of the plasma membrane is composed of proteins. It is these proteins that facilitate transport, among other things, of all of the molecules that are vital to the functioning of the cell. There are two major types of membrane transport proteins: transporters and channels. The major difference between the two is the way that they interact with the solid. Transporters undergo conformational changes and act sort of like a lock and key mechanism, so that the solute binding site is open first on one side of the plasma membrane and then on the other, while channel proteins form a pore that only certain types of molecules are able to navigate. Unsurprisingly, transport across channels occurs at a much faster rate than that across transporters. Another major difference in terms of the types of transport possible is passive versus active transport. Passive transport, otherwise known as facilitated diffusion, occurs whenever a molecule, or more often ion, is moving down its electrochemical gradient, which requires no additional energy to keep it going. However, some ions must move against their electrochemical gradient, for reasons which are beyond the scope of this video to address. In this case, the cell employs what is known as active transport, of which there are three types, and three major sources of energy which may be used by either type. The first and simplest type of active transport is a uniport, and a uniporter, in the case of the uniporter, molecules are simply pushed in one direction using one of the possible sources of energy, with the rate of transport simply being determined by the Vmax and Km of the transporter and solid in question. The other two types of active transporter are what are known as coupled transporters, which means that the passage of one solute is intimately connected to the passage of another. When the two solutes are moving in the same direction, the transporter is called a symporter, and when they're moving in opposite directions, the transporter is called an antiporter. With the co-transporters, one of the solutes is moving down its electrochemical gradient, which provides the energy necessary to move the second solute against its own electrochemical gradient. This brings us directly to the different sources of energy which are used in active transport. As mentioned previously, symporters and antiporters are the two types of coupled transporters, whose source of energy is the downhill movement of one of the solutes down its electrochemical gradient, which provides enough energy to push the other solute against its electrochemical gradient. The second type of energy used by transporters is ATP, where the transporter functions by coupling uphill transport to the hydrolysis of ATP. Finally, there are light-driven pumps, which basically couple uphill transport to light energy. These are found mainly in bacteria and archaea. Finally, channel proteins control what passes through them by having very particular conformations which only allow very particular molecules to pass, and by having gating mechanisms which are able to temporarily stop even the passage of molecules that the channel is designed to accept. This is accomplished in two major ways: the use of ligands and G-protein coupled receptors. Unfortunately, the more detailed mechanisms of action of these modulations are rather beyond the scope of this video. So, this is it. We hope that you have enjoyed our video. Thanks for watching.